Hi, my name is Felicia, and I am a PhD student in chemistry at KTH. And I'm going to give you a short introduction to the research project that I'm working with, which is called RADOV. And RADOV stands for Radiation Harvesting of Bioactive Peptides from Egg Proteins and their integration in advanced functional products. So the reason behind this project is the growing global health threat of antimicrobial resistance, which occurs when microbes such as bacteria no longer respond to antibiotics. So there's a great need to find a solution to this problem, and antimicrobial peptides, which are smaller proteins or part of proteins, might be a possible alternative. So in this project, we want to use ionizing radiation, which is high energy radiation, to cleave the chemical bonds within the protein chain so we can produce these antimicrobial peptides. We can then use them in different products such as hydrogel wound dressings or food packaging films. So one of the techniques that I use to analyze these samples is matrix-assisted laser desorption ionization, time-of-flight mass spectrometry. So we have our samples, which we will mix with a matrix. A laser will then ionize these molecules in our sample. The ions will then be separated in a mass analyzer based on their mass to charge ratio. They will then reach the detector and we will get our mass spectrum. So here's just a short clip from the lab. So we have our samples and we work with very small volumes. So this is only half a microliter that we will then spot onto our multi-target plates. So this is a great technique when we have very little sample or low concentration samples. We will then take our target plate and insert it into our instrument. So now the sample is going into the ion source or the moldy portion of the instrument. We can then select which spot we want to analyze, and we will see the sample matrix crystals here, yes. We press start and our laser will then start and we will start to record our mass spectrum. So here we see what ions we have in our samples. We have intensity on the y-axis and mass to charge ratio on the x-axis, which can be related to the size of our ions. That was all for me. I just want to thank European Union's Euratom Research and Training Program for funding this project. And I also want to thank you for your attention.